There's a section through the testis. Just got in a lecture, we put a long little tube called seminiferous tubules. That's what we're seeing here. Section through seminiferous tubules. It's in the walls of these seminiferous tubules that the process of sperm formation occurs. This is called spermatogenesis. We went over that already in lecture. I want you to know the cells involved. On the slides, I'm not going to ask you the individual cells involved in spermatogenesis. However, on the models, these bubble charts, I will. I expect you to pick them out. So what you can do is, if I point to one of these are the nuclei of cells that are directly involved in spermatogenesis. They're collectively called spermatogenic cells. They have kind of it looks like a granular nucleus because a lot of them are in the process of division. Sperm then are produced, released into the lumen, or cavity of these tubes, carried through a bunch of tubes on the way out of the body of the male. There is another, or two other cell types, actually three other cell types you should be familiar with. And here we see a cell with kind of an oval nucleus with a prominent nucleus. They tend to be more toward the outside of the wall of the seminiferous tubule. These are called sustentacular cells, or Sertoli cells. If you have your handout, we're not objecting to see this on page 16 of the testes under seminiferous tubules. Everybody see that? First we have the spermatogenic cells, and below that you have the sustentacular or Sertoli cells. These cells are not directly involved in spermatogenesis, but rather they produce nutrients to keep the sperm viable. So the sperm are living cells, like any cell in the body, it's got to be provided with nutrients. So again, these that have kind of an oval nucleus, you can see right here and right here is the prominent nucleus. These are the nuclei of the sustentacular or Sertoli cell. See one right here or right here as well. They tend to be more to the outside of the wall of the seminiferous tubule. The sperm or spermatozoa being here, we don't see it in this slide, but I'll show you something in a minute where you do see them. Sperm are pretty obvious because, of course, there's only cells that have what? Upper gelatin. All right, another cell type we see out here in the spaces between these seminiferous tubules, this is the interstitial spaces, so these are called interstitial cells or Leydig cells. This is also in your handout. Interstitial cells or Leydig cells are the cells that produce the male sex hormones, likely referred to as androgens. And the major one, of course, is testosterone. So these cells, again, are the interstitial cells or Leydig cells. They produce the male sex hormones, which, of course, are released not into the seminiferous tubule lumen, but into the blood. What hormone stimulates these cells to produce male sex hormones? Pituitary hormone. Which one? Luteinizing? Yes, luteinizing hormone. Which is why in males, luteinizing hormone is also known as interstitial cell stimulating hormone. It stimulates these cells to produce the male sex hormones. So you should be able to pick these out. Also nuclei of these cells, the sustentacular cells, and then collectively the cells that are directly involved in spermatogenesis, the spermatogenic cells. Okay. And of course the sperm cells you can see are in the lumen, but we don't see them on this slide. Something else you're going to see on this is the capsule. This connective tissue membrane that surrounds the organ has a fancy name. It's called the tunica albuginea. The tunica albuginea. It's also in here. Tunica albuginea. And under testes, you can see tunica albuginea. Okay. Now most of the slides in the collection are going to look like this. Again, you're going to see a lot of sections through seminiferous tubules. You can see here. Again, these cells, most of the cells again are going to be spermatogenic cells. Look at the nuclei of spermatogenic cells. Again, they have kind of a granular type nucleus. These are cells directly involved again in sperm cell production. You also got cells in between the seminiferous tubules. These would be the interstitial cells, again, that produce the male sex hormones. Interstitial cells or the Leydig cells, those are also known. And as you go through, if you look to the outside, you'll see some of these oval nuclei with the prominent nucleolus. These again are the nuclei of the substentacular or sertoli cells that provide nutrients for the sperm. And again, on the very outside, the testes. You can see again, connective tissue membrane that surrounds, supports them. The capsule is again is the tubular alligator. Tubular beginning, and again, these are seminiferous tubules. And in between the interstitial cells. This is section to the human testis. What we saw before was actually the rat testis. And here again, another section through testis and the rat. But this one I want you to see because it shows something we didn't see in that first slide. Sperm cells. You can see them here. It looks like hair is extending into the lumen, seminiferous tubule. Those, of course, are the flagella of the sperm cell. In the case in rat sperm, the head of the sperm looks a little different than it does in humans. It sort of looks like a little hook, like a fish hook without a barb. Again, they're flagellated just like human sperm. They're produced by these cytogenic cells that release the lumen of the seminiferous tubules of the testes. And again, you can see here's part of the capsule, the tunica albuginea, that surrounds the organ. Any questions on the testes? Sperm then passes from the testis to a series of tubes, which I don't expect you to know the names of, but eventually it's picked up by tubes in the next organ, which lies alongside of it. And this is the epididymis. The epididymis also has a lot of tubes within it. Sperm again are in the lumen of these tubes. See them here. This big mass of cells here, this is the sperm by the millions. Now, if we look at the lining, the epithelium that lines the wall of these tubes in the epididymis, you should know the type of epithelium looks much like we saw in the trachea, but not quite. Like the tracheal lining, it was what kind of cells? What are these?
Pseudostratified.
Pseudostratified columnar. But unlike the trachea, which had ciliated pseudostratified columnar epithelium, this, even though it appears to be ciliated, is not. What we see here that appear to be cilia are actually what are called stereocilia, and they're really microtubules. Excuse me, they're really microvilli. Their function is not to move the sperm like cilia would do. It's to provide nutrients for the sperm and to absorb excess water from this mass. Again, the sperm are going to be in the body of the male for a while. You've got to keep them viable by providing the nutrients. So again, these are microvilli, stereocilia for the sperm, not for moving the sperm, but to provide nutrients and to absorb excess water. So you can see a sperm cell right here in the flagella. So again, the epithelium is pseudostratified columnar epithelium. but not ciliated, as it would appear. What moves the sperm, they're not swimming at this point, contraction of the smooth muscle in the wall of these tubes. Now the sperm member are temporarily stored here. What else occurs in the epididymis?
Maturation.
Maturation. So when the sperm leave the testes where they're produced, they're capable of fertilizing an egg. They are not mature yet. The maturation process occurs here. And usually they're not swimming, and they usually don't start swimming until they're released from the body of the males, for saving their strength, if you will. They're moved along passively by muscle contraction in these tubes. Also, just external, or deep, if you will, to this epithelial layer, again, we got loose connective tissue. This is a mucous membrane, so what is this connective tissue called?
It's lamina propria.
Lamina propria, like we saw on the walls of the GI tract, also in tubes of the urinary tract as well. So pseudostratified columnar epithelium, not ciliated, and the lamina propria, and the muscle layers. And there are three muscle layers. It's all smooth muscle. So the osteotypic tissue is smooth muscle. The arrangement differs. Inner, longitudinal, middle, circular, and outer, also longitudinal. There's longitudinal right here as well. You got three layers of smooth muscle. Okay? Here we get longitudinal, the circular, and the longitudinal. Like we saw in the wall of the urinary bladder. Okay, so it's like the urinary tract, tubes. Inner longitudinal, middle circular, outer longitudinal. Again, there's the epithelium, pseudostratified columnar, lamina propria, there's the muscle layer, longitudinal smooth muscle, there's the circular smooth muscle, and there's a longitudinal smooth muscle. Okay? And again, if you get a pointer on this, don't give it the whole wall, just what it is. So it's out here, you can see the edge of the tube or outer part of the tube, it's longitudinal. It's here in the middle with the muscle on either side, it's circular, and it's next to the lamina propria. Okay, you can actually see the epithelium right in here, and again would be longitudinal. So type of tissue, smooth muscle, arrangement, longitudinal, circular, longitudinal again. Again, it's a contract that muscle that moves the sperm, not the stereocilia, which are microvilli. Any questions on the difference? Again, function, stored sperm temporarily, site of sperm cell maturation. All right, then there's a tube that carries the sperm from the scrotum where it connects to the tubes of the epididymis, up in the body cavity, runs around behind the urinary bladder, penetrates the prostate gland, and joins the prostate urethra. That tube is called the ductus deferens or the vas deferens. Now be careful because a lot of slides are labeled spermatic cord. And the spermatic cord and the ductus deferens are not one and the same. Think of spermatic cord like a cable and a lot of tubes within it or wires within it. One of which is the tube that actually carries the sperm, which we see here, and this is the ductus deferens or vas deferens. Again, it's taken from the scrotum, where it connects to the epididymis, up in the body cavity, where it connects to the prostate urethra running through the prostate gland, just inferior to the urinary bladder. Again, we see the same layers in the wall that we did before, in the epididymis tubes. You got your pseudostratified columnar epithelium. Sperm would be in here. Pseudostratified columnar epithelium, like we saw before, not ciliated, though it appears to be. And then, again, you got your layer of lamina propria, like we saw in the tubes of the epididymis. This is lamina propria here. And then the same three muscle layers, same arrangement. Again, so you got your longitudinal smooth muscle, and the circular smooth muscle. And again, on the outside, longitudinal again. See perhaps a little better even with lower magnification. You can see it actually even much better when you look at it through the microscope. Here again, here's the epithelium, pseudostratified columnar. Here's a little bit of the lamina propria. That's the longitudinal muscle layer. That's the circular muscle layer, and that's the longitudinal muscle layer. So type of tissue, smooth muscle again, arrangements, longitudinal, circular, longitudinal. And again, I'm giving the whole wall, just like the corners on. It's up here, that's the lamina propria and the epithelial lining. It's longitudinal. In the middle, we got muscle on either side. It's circular. On the outside, and longitudinal. Again, this is the tube that carries the sperm from the scrotum and connects with the prostatic urethra as it runs through the prostate. And by the way, this is what's cut when a male is sterilized. You usually don't cut in the body cavity, you cut it down basically the scrotum. You cut the tube, tie off the ends, that's called a vasectomy. You're tying off, if you will, the vas deferens or ductus deferens. What that does is obviously prevent sperm from getting in semen, so it basically sterilizes the male. But it takes a while. Overnight, because you're going to have some residual sperm in the tube, that's why they say, you know, be careful to prevent conception. Basically use other means for a while or two to make sure all the sperm is clear in the tubes of your reproductive tract. The slide is labeled chromatic core, so be careful because it's chromatic core.